Hello, everybody. Again, it's my second time this week to have the pleasure of reintroducing Willi Gruissem. You've been here yesterday, so you know all about his achievements. I'm not going to go over his achievement, otherwise the whole hour will be taken. I'm going to tell you about his basic science. Yesterday, he talked about the translational science that he's doing towards improvement of cassava and rice, using the basic tools that he has developed in his lab, using the systems approaches for quantitative analysis of plant growth and development. Specifically, his research programs are targeting many areas. I just picked up few. Among them is the retinoblastoma-related proteins, RBR proteins, known to be involved in cell division and differentiation. He has expanded that work, and he has shown that any mutation in RBR genes results to gametophytic lethality of the plants. And now he has developed that even further to show that this group of proteins interact with the protein involved present in a chromatin complex. And now they are developing again tools to understand how the developmental processes from vegetative to reproductive system are controlled. So that is one of his major areas. The other major area that he's tackling is functional genomics. So we talked about, or I mentioned about Genome-Scale, that most of you perhaps have already used it, because it's free for academia to use that system in order to look at the genome-wide expression profile of the plant and animal genes in that database that they have developed. So in addition to that, he's looking at mass analysis, mass spectrometry analysis of the proteins present in Arabidopsis. They have sequenced already 14,000 proteins, and now he is correlating the expression of the genes with the translational machinery in order to decipher regulatory mechanism involved in translational, transcriptional, and post-transcriptional mechanisms. The other part that he is working on, the other project, is on the isopronate pathway, a pathway regulated by plant stress responses and also developmental responses. He wants to understand the pathway, how is regulated, and how this pathway regulates other metabolic pathway in response to developmental and environmental cues. And lastly, he's looking at taking all this knowledge in order to develop plant biotechnology to apply to cassava and rice, the project that he talked about yesterday. Today, however, he's going to focus his talk on genome-scale proteome and gene expression exploration of chloroplast development and metabolic networks. Willi, thanks very much for coming. It's a pleasure to have you, and podium is yours.
Katie, thanks a lot. And again, welcome everybody. And
oh yeah, I forgot.
Thank you very much for inviting me, having the opportunity actually to give a second lecture here and discuss with you a little bit more about the work that we are doing at the ETH in Zurich. So Katie already gave away my talk. But yesterday I spent a little bit of time talking about genetic engineering of plants. I mean, obviously, we like to engineer plants for specific traits to make plants, higher yielding, more drought tolerant, resisting cold and whatsoever. But ultimately, we need to understand the underlying mechanisms. What is it actually that confers certain traits, and unraveling this network of genes and mechanisms is really ultimately what I think will drive breeding in the future. So what I would like to do today is discuss with you sort of our first timid approaches to understand the plant as a system. I wouldn't call it yet systems biology, but we are certainly trying to take systematic approaches to understand plant function. I mean, one project that we are engaged in right now that I will not talk about well, we are part of a large European consortium of about 12 labs, and we are all together focusing on Arabidopsis leaf number 6. So hopefully in 2 or 3 years we have better understanding what actually leaf development means in molecular terms and in terms of genetic networks. So it's actually a very, very interesting project, because for the first time I'm involved in experiments that have to be coordinated between different labs. And tell you, it's not easy. It's not easy, you know, just to make sure that everybody grows plants under the same conditions. You know, we did that first experiment to try it out, and just to give you an idea, the paper is almost ready, so I can talk about that. But of 10 labs, you know, only 4 were able to sort of hit the median, even though we all use the same protocol. Okay, so that's good that 4 labs were able to do it, but it also means it's very difficult to follow a standard operating protocol. Nevertheless, so again, then systems biology, of course, is sort of the buzzword that everybody is using. And for me, what does systems biology mean? You know, there's as many definitions as there are scientists around that work in this area. So I asked the experts, and of course the key here is to understand the interactions between components of biological systems and how these components, these interactions, can give rise to function and behavior. So that's the idea. And of course, you break it down then into the into the different parts of the system. And of course, mostly we're trying to understand the genetic, biochemical, and metabolic networks, because they together really then drive the functions. And in a way to understand this is you set perturbations, and then these perturbations give responses, and then hopefully from these responses then you can infer the cause of the perturbation, or what actually you have perturbed, and mechanistically how this works. And in order to do this, you need to understand, or you need to measure, quantitatively measure the system's responses. And we now have the tools to do that, both at the RNA, the protein, and the metabolite level. And even then, I mean, once you have quantitatively measured the systems, I mean, I personally would not say you understand the systems. You do systems biology, but the best we can do at the moment really is to infer causality. Okay, because there's many other parameters that need to come into the equation, such as enzyme concentration, enzyme kinetics, and metabolite concentration. Some of those we can measure, but by no means are we able to really measure all of the parameters at the present time, and certainly not necessarily, I mean, if we talk about the entire plant, not necessarily in a cell-specific way. And then in the end, what you need to do if you really want to do modeling, and what we are working on right now, I won't have time to discuss it today, is when you have all these different types of data, you know, so of course you analyze each data set alone, but ultimately you need to bring all these different types of data together in order to do modeling. And this is we have a large project actually going on in Switzerland right now. It's called systemsx.ch, where we are working with different labs and a number of specialists in bioinformatics and data processing and data integration to build a pipeline that allows us then to integrate various types of data in order to do modeling. And then, of course, the modeling, and again, I mean, I'm not an expert there. I'm learning right now. It's actually a lot of fun to see what kind of modeling you can do. I mean, most of the modeling, to be honest, that we are doing today is this top-down modeling, where you have a lot of data, and based on the data you infer some kind of networks. And with a few genes, of course, you can also do this what we call bottom-up modeling, where you develop models and then you test the models in your biological experiments. Now, the types of models that you can describe right now based on the data sets that you have available are either these, what we call interaction-based model or constraint-based model and mechanism-based models. And in our lab, we are actually pursuing all three types of models depending on the types of data that we deal with. And in fact, we have just started a new project based on the protein and RNA data that we have to do constraint-based modeling on trying to understand what components of metabolic pathways we are actually still missing, and that you can, and these would come out of these types of constraint-based models. Well, data integration actually means different things to different scientists. We look at data integration first of all from the data types that we have, transcript, protein, metabolites, interactions, phenotypes. So all of these have to come together. I mean, we actually tried that. You can't do it in Arabidopsis right now or in any other multicellular organism, but we tried that in yeast, where we used all the available data types that are currently available for yeast and tried to integrate these data types to predict synthetic lethality. So which genes are actually interact with each other. We have the paper submitted to PLOS Biology right now. Hope, of course, that they will take it. And then what is interesting, not every data type works for that, but there are certain data types, like, for example, transcriptome, proteome data that work quite well, and that has allowed us to predict at least 30, 40 different new genes that are probably central nodes, and that can be then experimentally tested. Okay, so the whole idea is that from this modeling, that you develop testable hypotheses that you then can follow up again in experiments. But in order to get there, of course, you have to generate the data. That is in itself a lot of work. We have done that in Arabidopsis, so this work was published last year, where we have tried to identify as much of the Arabidopsis proteome as possible, and we are doing this using different types, different parts of the Arabidopsis plant, and as Katie already pointed out, we have, this is an old slide already, it's outdated, identified more than 14,000 proteins, but actually the number is higher now. We are pushing 17 now because there are a few other organs that we have included. And you see that these are based, that this information is based on hundreds of thousands of spectra that we have derived, and from which we are predicting many types of peptides, and from these peptides and predicting the proteins. And we are very stringent in our analysis, so you have to identify at least 3 or 4 peptides for a protein before you consider it identified. But it's been a very dedicated team of colleagues who have been pushing this work. So what has come out of this? What can you actually do with this type of analysis? Well, first of all, it's helped us to improve the Arabidopsis genome data. Okay, so you can actually go in there, and often what we see is that we find peptides that correct the gene. For example, here is a gene that has been annotated, but we're seeing peptides here. So presumably that gene is actually much longer, okay, than what the annotation predicts. We are correcting gene models also, so there are a number of things we can do, and you can read it up in the paper. And in fact, there is another study out right now from Steve Briggs's lab, where they've used another analysis software than we are using, and they're finding actually a lot of small open reading frames in the genome that we have not considered at this point, and we are using their data now and our software, and we're running through this again and confirming each other's data, which is actually also very good. All this information is now in a database that you can access. So if you go to atproteome.ethz.ch and type in your favorite protein or proteins, your protein will come up. So it's right there, the entire protein. But what is more interesting then is that based on the information that we have, so based on all the data that are in the database right now, you can actually paint your developmental or your organ-specific landscape as you wish. So we have here just shown one example where we are comparing the protein landscape of flowers, leaves, roots, seeds, calyx, and tissue culture cells. And you see that there's really a unique signature to each organ or tissue that we are using here. And this signature, this protein signature, is predictive for the function of the organ as well. So you can actually play around with that and learn more about the biochemical networks and also the signal transduction networks that are functional in these organs. Now, of course, we don't have all the proteins yet, so the networks or the landscape is still pretty patchy. We also don't know yet, how many more proteins we have to find in order to fill up. Of course, you can go by the gene prediction of the genome, but that doesn't necessarily mean that all the genes that are currently predicted in the genome encode functional proteins. And I will get back to that in the end. What I think is actually more important, and what I think will be very useful for the community in the future, is the information that is in the database on specific peptides. So if you take a particular protein and you isolate that protein from different tissues, so say for example it's a protein that is expressed very widely in the plant, and you do mass spectrometry analysis on these different protein fractions, you don't always see the same peptides of this protein coming up in different tissues. We don't understand yet the mechanism behind it, but these specific peptides that come up in certain tissues, you can actually call the signature of that protein in this particular tissue. And based on, we call them proteotypic peptides, and based on these proteotypic peptides, you can actually do quantitative proteomics. So you can use these specific peptides to specifically look for the expression of your protein in a particular tissue. And that's been very useful, and there's a lot of interest in the field actually to use these proteotypic peptides as certain biomarkers to look for the expression of these of certain proteins that you're interested in. So you don't have to do Westerns or so, but you can take a labeled peptide, spike it into your extract, and then tell the mass spec to specifically look for this protein. And since you know how much of your labeled peptide you put in, you immediately get quantitative information. So it's a very, very powerful approach. So in order to build this database, we had to actually use quite a number of analysis algorithms that have come together. So I just give you an idea, that it's not a simple program that you run and then you get all the data. You actually have to combine a number of different analysis tools in order to come to this database that we are building. And this database, in the end, we will also try to merge with the gene expression data that we have. I'll show you at the end what we are doing there to give you an idea how you can then compare transcription and protein accumulation. All the data that we have currently from the proteome, we have it in the AT-proteome database, which is a little bit more dynamic, so you can work with that quite easily and customize your searches and your analysis. But all of the information is also in this public database called PRITE. And in the public database, what we have also done, we have included the actual mass spectrometry data. So you can not only go in there and get the peptide information or protein quantity, but you can actually download the spectrum itself and reanalyze and make sure if we made the right predictions. So there might be other experts out there that are better than us and may correct our data in the future. I hope we are right. But again, so if you go into the AT proteome database, show you just a couple of examples based on the peptides that we have identified. You can go into the database now and get information on all the different peptides that are available. Okay, so that is very powerful. If you want to analyze your own proteome, you can customize these searches and get information on your proteins that you are interested in. We are also giving you quantitative information. So because the mass spec analysis that we have done is all done in biological replicas, many technical replicas, and so on. We have used, oops, sorry, that was too fast. We have used an algorithm that was developed a couple of years ago and adapted it to the Genome-Scale data. So this algorithm was actually developed for a small-scale proteome analysis in yeast, and we adapted this algorithm to the Arabidopsis proteome data. And based on this algorithm, we simply quantify the spectral information that we get, and then we put this quantitative information into the database so that you can actually go in there and get quantitative information on the accumulation of these proteins in specific tissues. Okay, so you have basically your Western blot analysis in the database. So that gives you a very good indication to what level these proteins are actually expressed in Arabidopsis in the various tissues and organs. So then, just to give you sort of also an example of how you can drive this analysis further and how you actually dig deeper into the genome, you can sometimes use a few tricks. I mean, if you're familiar with the constraints of mass spectrometry, you know that very abundant proteins often mask lower abundant proteins. So it's a problem of dynamic range. And to illustrate that, I want to focus in the next few slides on the chloroplast, because although we have identified many proteins now to be associated with the chloroplast, 1,633. We've also done similar analysis for mitochondria, peroxisomes, and so on. To the phosphoproteins, I will get back to that in a few minutes. But the question that we are begging to ask is, I mean, how many proteins are actually in the chloroplast? What constitutes a proteome of the organelle? Right? And how do you actually do that? Because if you go into chloroplasts, well, then your high abundant proteins are, Rubisco, the LHCPs, and you find them again and again, and it doesn't help you to track down lower abundant proteins. So, and to show that is that, if you look at all the proteome studies that have been done for the chloroplast, even looking at subsections or subfractions of the chloroplast, the number has been, based on the literature. I mean, a lot of it is, of course, our work, but there are many other labs that have contributed to this. The number has been sort of hovering around 1,400. I mean, the 1,600 is actually the more recent number now that we have. So how do you push it further? Okay, how do you actually know what is in the chloroplast? So the problem is that the analysis tools that we have for predicting chloroplast proteins are not very sensitive. So, for example, Target P, which is sort of the program that most labs use for the prediction of plastic proteins, the sensitivity is only about 75% if you look at non-photosynthetic plastids, and it even drops down if you look at chloroplast. So the tools are not very sensitive. What can you actually do to improve the sensitivity of the tools? Well, of course, the problem is that there might be alternative pathways. So Target P predicts mostly proteins that have transit peptides, okay? But there might be alternative pathways by which proteins get imported into the chloroplast. And then, of course, you will miss them with your prediction program. So the only way to identify these proteins is actually by biochemical means. The computer prediction programs are not helpful. And in fact, in the last few years, there have been a number of papers now that suggested that in addition to the sort of canonical chloroplast import pathways where proteins are recognized by receptors at the surface of the membrane and then imported into the chloroplast, there might actually be alternative pathways through the secretory pathway by which proteins go into the chloroplast. So you would miss them by using the computer prediction programs. Well, a smart graduate student in the lab, Sylvain Bischoff, actually came up with the idea and said, well, why don't we look in chloroplast import mutants that are unable to import photosynthetic proteins. So you know that the import apparatus consists of many proteins. So the one that is important for import, the TOC159, when you knock this out, you get actually pale plants. Okay, so you cannot grow these plants in soil. You have to grow them on sucrose because they are unable to import photosynthetic protein. Another import mutant is PPI1. PPI1 is this TOC33. Where is it actually? Well, it's actually TOC34, but it's a little bit of confusion about how this protein should be labeled. Here it's labeled TOC34, but it's TOC33. And this protein also participates in the import, but it doesn't seem to be so important because in this case you get import also of photosynthetic proteins, but it is compromised, okay? And then this is actually the PPI2 mutant complemented with another member of this TOC159 gene family. So he actually started to do proteomics on the chloroplast of these mutants because the rationale was, well, if we're not having all these abundant photosynthesis proteins, we should be able to detect additional proteins that are imported into the chloroplast. And the answer is yes, he did actually identify them. So the current number of proteins that we have identified in plastid is pushing around 2,700. Okay? So he has identified many more low abundant proteins by simply taking out these high abundant proteins using the import mutant. So I thought that was actually a pretty clever approach. Now, what are we finding here? Just to give you a little bit of background, so these are all unpublished data. We are finding that in the PPI2 mutant, we actually have proteins enriched that don't have the canonical chloroplast import sequence. So that is something that you would expect. And you see that these proteins are actually missing from the wild type because you have these abundant class of photosynthesis protein. Okay? So I'm not going to go through all the details, but we can then ask the question, what are these proteins? In which particular class do they fall? You can use statistical analysis to look for protein which are more abundant in wild type than the PPI1 mutant or the more abundant in the PPI2 mutant. And when you then relate this to transit peptides, you see that in the wild type or even in the PPI2 mutant or even in the complemented mutants, you see that most of the proteins or many of the proteins we are finding have certain signatures in the N-terminal region of the protein, whereas you're not finding these signatures, and Steve, we can discuss it in more detail. I think it's actually very interesting. We don't find that particular signatures in the PPI2 mutant, so there's no clear enrichment for a particular class of transit peptides. Then, of course, what you need to do is you need to confirm that these proteins, that these additional proteins that you identify are actually localized in the plastid, that you have not co-purified some non-chloroplast proteins. And of course, we can't do this for every protein, but for a number of examples that we picked, we made GFP fusions, and for all of them, we have been able to confirm that they are actually in the chloroplast. So we are quite confident that, I would say, at least most of the proteins, additional proteins that we have identified, are chloroplast proteins. So that pushes now 2,700 proteins, which is getting close to what is being predicted for the chloroplast, 3,000 to 3,500. Okay? We know we are still missing a number of very low abundant proteins that have been identified in targeted biochemical assays that we don't have yet in our proteomics approach. What was also interesting, what came out of this, when we specifically looked at the import machinery itself in these mutants, I was actually a little bit surprised, because I would have expected that if you take parts of the import machinery out of this complex, that the import machinery would fall apart. But this is not what we are seeing. So we are not seeing, if you look at the various components of the import machinery, in the mutants, we are not seeing a sort of a large-scale reorganization of the import machinery, which is very interesting. So here is simply the proteins that we have identified, and the different color here indicates their abundance. So we quantified the proteins, and so you see that the abundance of these proteins in the different mutants is the ones that we detected, which have a color here, is very similar. So there's not a large-scale reorganization of the actual import machinery if components are missing. But what you also can see, if you, again, we are building a database to specifically mark these proteins. You see that, and you see this here very nicely, if you look at specific photosynthetic proteins, they are clearly overrepresented in the wild type and in the PPI1 mutant, but not in the PPI2 mutant. So we have clearly, I mean, we can show this simply by statistical analysis here, that we have depleted these proteins. Now, with all of this information, what becomes nice then is that you can do correlation analysis and actually learn more about the function of these organelles. And I just give you one slide here to illustrate this point, where we have done a correlation analysis for all the proteins that we have identified in mitochondria and plastids and in peroxisomes. And you see that there's not really a very strong correlation for mitochondria. That is, that the proteome of mitochondria is very similar in different tissues. Okay, so there's no specificity of the particular protein for a particular tissue. But when you look at plastids, you see that the correlation clearly diverges here. That you have clearly a photosynthetic proteome that you find in leaves and shoots and flowers, a little bit in calyx, but then in roots you have a completely different proteome. Okay? And for peroxisomes, the situation is very similar. Okay? So that gives you an idea that the proteomes of these organelles, if you look at it at the genome scale level, so look at all the proteins, that these organelles clearly have very specific functions in different tissues. Okay? So you can then, of course, go in and break it down to the individual proteins and pathways. I'm not going to show that here. But starting with this large-scale analysis, you get then an information on the function of these organelles. You can combine that with transcription data. Here is transcription data from about 2,600 Arabidopsis gene expression arrays, and correlate this with a protein accumulation. And then what comes out of this, you won't see this very well here, but we have color marked this. You get actually co-expression of certain classes of genes across organelle boundaries. So that is that certain classes of genes are regulated with the organelles rather than with individual organelle types, or with developmental programs, or even with environmental effects. But they're clearly regulated with the different organelles. So you can do this analysis very nicely. I come back to that in the end of my talk. And then just a word to the phosphoproteome. Again, I'll give you one example here. That's a dedicated graduate student who has initiated this work, trying to understand the Arabidopsis phosphoproteome. And she first sort of developed these techniques to isolate phosphorylated proteins, enrich phosphorylated protein using Arabidopsis cell extracts. But then to drive a biological question, she actually asked, how are the phosphoproteomes, how do they differ at the end of the day and at the end of the night in the plant, or in the leaf specifically? And so she isolated the phosphorylated protein, or the enriched phosphorylated proteins, and analyzed them by mass spec. And then I'm specifically focusing just on the analysis of phosphorylation motifs for chloroplast proteins. Now I'm going to ignore all the other about 1,600 phosphorylated proteins that she has identified. I just want to give you one example of what you can do when you have that information for the chloroplast. So one thing you can do is take the information from the peptides that you find, from the phosphorylated peptides that you have enriched, and ask, is there actually a specific motif that comes out of this analysis? And here you see that when you align plastidic phosphorylation site, that there's a serine and threonine that is very dominant. Okay, and you can then drive this information further and ask, are there actually different types of signatures, and how do these signatures relate to what we know about specific kinases? And when you do this analysis, you see that there are three major signatures that come up in the chloroplast. There's a kinase that has actually not been discovered yet. It's what we think a C-terminal directed kinase that gives you this specific signature here. It's a proline directed kinase, and then there's a casein kinase 2 signature that comes out of the alignment of all these phosphorylated peptide. And for the casein kinase, so that paper is submitted now, we have actually been able to predict what we call a casein kinase 2 network, where the arrows indicate the degree of significance that we think for these different proteins that are phosphorylated by casein kinase 2. So you see, we have identified quite a number of proteins. I mean, there's actually classes of proteins here that we have identified in transcriptional regulation, post-transcriptional regulation, and metabolic regulation, as well as some of the photosynthetic proteins that appear to be phosphorylated by casein kinase 2. And that is very interesting because what this kinase seems to be doing is connecting metabolism and energy metabolism with transcriptional, with gene regulation. Okay? So it sits in a very, very central node by phosphorylating different types of proteins. And of course, it's very, very interesting now to ask the question, well, where does casein kinase 2, where does it get its input from? So what regulates this particular kinase so that it then connects a metabolism with gene regulation? So you see, I mean, based on this kind of information, you can then ask the next level of interesting biological questions. So in the last few minutes, I want to show you the effort that we are making to bring this protein and transcript information together, and then ask what can we actually learn if you have quantitative information on genome scale, quantitative protein information, and transcript information. And to do this, I mean, well, we're using now Genevestigator to do that, but this is not how Genevestigator actually started. So the person who was driving Genevestigator, the development of Genevestigator, is Philip Zimmermann. I don't want to spend a lot of time sort of explaining the underlying rationale for how Genevestigator works, but just to let you know that we are taking the information, the gene expression information that are out there, and then the work that we are doing, which is very important, is to annotate this information, so that with the annotation we can then actually develop a database that allows you to pull out specific information. And therefore for us it's actually very important when you do gene expression experiment that you annotate your experiments as carefully and as complete as possible. That helps a lot. That helps us a lot to do this type of analysis. And then it's also data quality control that is very important. So we are not putting just anything into the database. I've often been asked by colleagues, why are not my gene expression data not in the database? Well, the simple answer is that they haven't passed the data quality control. There was something wrong with the data. You know, it didn't fit the statistical evaluation. And so then combining the annotation with the data, and after the quality control, you then get a database that allows you to mine the database. And what we have now, in fact, that is an old slide I just realized earlier this morning, because we have now also built a rice database. We are working on a soy database. We're working on Drosophila. We're working on maize. And of course, the other databases are not as complete as Arabidopsis. Arabidopsis, I think that number is actually pushing 3,500. So there are 3,500 individual experiments from Arabidopsis in this database. I think the best one right now is human. So human has the most experiment. And it's actually very interesting with this database now. What it allows you to do, you can, for example, do your experiment in Arabidopsis. You look for your favorite genes. Okay? But then you can also correlate the analysis of these genes if there are similar experiments in barley and in rice across different plants. And if these are genes that are conserved in humans, mouse, and rat, you can even go into the animal background and look how do these genes, or how does the expression of these genes actually behave in different organisms. Well, when we originally published Genevestigator, it was, I think, the end of 2004. I don't know if that's the latest number, but it's been, I mean, the database has been cited quite frequently. I mean, the number is not so important here. But what is important is that all of these citations are actually, or most of them are actually correlating, or at least in the beginning, they have correlated the information from Genevestigator with the own analysis. So you have done real-time PCR, you have looked at promoter gas fusions, and to show that there is a correlation between the data from the database and your own experiments. And that has been extremely, extremely helpful because I know of only one publication where there was a divergence for one gene. Okay? Otherwise, the correlation has always been very good. And in fact, I mean, the journals have now gone, you don't have to do these control experiments anymore. If you go into Genevestigator, you take Genevestigator data and you publish them, they don't ask you to do controls anymore. Okay? So because of the reliability of the data in that database. So I find that that is, for me, what was the important aspect, that we have built a database that is high quality and reliable. So what can you do with this? Just one example here. I've played around a little bit. If you go into AT-proteome, you ask the AT-proteome, give me the top 100 seed-specific proteins. And then the AT-proteome database gives you this information. Okay? So here you have these proteins. You have the gene number here, so you know which genes they are. And then you go into Genevestigator and ask, well, are these top 100 seed-specific proteins really seed-specifically expressed? And the answer is yes. So that's very nice. There's a very good correlation between the AT proteome and the Geninvestigator database. And so you can actually play around with that. I mean, you can get information. You can pull information out together for your genes and your proteins. We have done this actually on a genome scale. So we have correlated all the protein data that we have with all the transcription data, so that is all the genes that correspond to these proteins. And what do you get then? So what you see is actually very interesting. You don't get a straight line that you would expect to get if there's a strict correlation between the expression of the gene and the accumulation of the protein. So in other words, if the protein is under strict transcriptional regulation, then you would expect to see that there's a strict correlation also between the accumulation of the RNA and the accumulation of the protein. That's not what we're seeing. So you can go into in all the tissues that we have analyzed, you always get this cloud. Well, what that cloud means is that, yes, there are many proteins that are transcriptionally regulated, but there is also proteins that fall out of this regulation. Okay? So there is a lot of post-translational, post-transcriptional regulation going on in Arabidopsis as well. So you can then, I mean, if you do this, of course, at a genome scale level, I mean, you get this cloud, and that doesn't tell you very much. Okay? It just tells you it's not strictly transcriptional regulation what's going on there, but you want to break it down and get more detailed information. You can actually do this, and here we have picked, in this example, I think we have picked about 400 or 500 genes from various metabolic pathways, and I just want to show you a few examples here where we have correlated transcript abundance and protein abundance. And so what you see here in the red area is where you have more protein than transcript, and here in the green area where you have more transcript than protein. So you see that within a certain range, most of the proteins fall onto the line. So that's very comforting, because it tells you, yes, most of the proteins are under transcriptional regulation. But then some of the proteins are not. Okay? And then you can ask, well, what are these proteins? And then you can go specifically, you break it down into specific, to the individual pathways. And here are just 4 or 5 different pathways that I've shown. When it's black, it means there's a good correlation between transcription and protein accumulation. When it's green, you know that there's more transcript than protein. When it's red, there's more protein than transcript. And you go to every single pathway, you see that for most of the enzymes in these pathways, there's a good correlation. But for some of the enzymes, it falls out. Okay? So what are these enzymes? And you can do this analysis. You can go pathway by pathway, and you see Kelvin cycle. Most of the enzymes are under transcriptional regulation. Some of the enzymes are not. So what are these enzymes? Why is it glyceraldehyde 3-phosphate dehydrogenase? Okay? Go starch and sucrose metabolism, fatty acid biosynthesis. So you can go pathway after pathway. You find genes that are not, or find proteins that are not under strict transcriptional control. I mean, of course, we asked ourselves the question immediately, well, is there something specific about these proteins? I mean, maybe these proteins biochemically are very difficult to extract, and therefore we don't see them in the mass spec analysis. Okay, but I mean, believe me, we have done so many experiments, and we have checked, we have done the controls, that we are fairly certain that this is not an artifact of the protein, that this is not a problem of the biochemistry that we are doing, but that this is really something that has to do with the regulation of these pathways. Here's one example out of the Kelvin cycle, and I find that actually quite interesting, because what it suggests here that there are really specific types of enzymes that fall into these groups of proteins where the strict transcriptional regulation breaks down. And often it's also various isozymes from this same gene family that, that fall into this pattern. Okay? And one last example here, fatty acid biosynthesis, so acyl CoA oxidase. So you have 3 isozymes here. They follow a very similar pattern. Okay? So now that of course raises a number of questions that I think will be very important to address in the longer term in systems biology. So if you look at metabolic regulation, we always assume that all the enzymes are there, and then that there's a flux of the pathways. I mean, of course, it's regulated perhaps by phosphorylation or so, but we have not really yet appreciated that a lot of the regulation may actually also occur at the level of protein availability, enzyme availability. Okay, so we don't understand yet how these mechanisms are regulated. But I think this type of analysis opens up a new area of questions and research opportunities, I think. You can even do this in much more detail here. So here, for example, we have used 4 different proteins. We have just followed these proteins in different tissues or in different organs. And you see that again, some proteins, I don't want to dwell on this now, but some proteins really show low protein to transcript ratios in all tissues. Some proteins always show a good correlation, and some proteins always show a high protein to transcript ratio in all tissues when we've looked at that. Okay? So why is that? Okay, so you really need to go in now to these individual proteins and ask questions. You know, how are these proteins, or how are the genes for these proteins, how are they regulated? And this type of analysis can help you to identify these proteins that then you can analyze in more detail. So when we thought, you know, biology was over, I mean, there's a whole new area that is opening up here that I think we will have to address in the future, especially if we want to understand how the system actually works, you know, because I think that will actually have also implications for, as I mentioned earlier, for the breeding efforts. Then the last point that I just wanted to bring up here is something what we call extreme proteome transcriptome discrepancy. So often when you do proteomics, it's very difficult because of the dynamic range and the number of other parameters to find all the proteins. But when we again compare the proteins that we have identified and the proteins that we have not identified that you can predict based on the genome annotation, and we correlate that with transcript abundance, then you get specific curves. That's what you would expect. Okay, but why is it that certain proteins, if the transcript is very low abundant, we don't see, but others we do? Okay, so you hear at the fringe, and again, on the other hand, transcripts are very abundant, but there are still certain proteins we cannot find, okay, even though their transcripts are very abundant. So I showed you a few specific examples earlier where I said, well, you can go to specific parts where you see that they're individual proteins, but then if you do that analysis at the genome scale level and ask, in what categories do these proteins actually fall, you see that so we're using these gene ontology categories here. They're not perfect, okay? But what is interesting is that you see that these proteins actually fall into specific classes. So it's not a random distribution of proteins here, but it is very specific classes of proteins in which these extremities fall. Just one example here is translation. Well, that part we can explain very easily. So you don't detect these proteins. Well, that's not true. We are detecting them. These are ribosomal proteins, but ribosomal proteins are multi-gene families. These proteins are so similar that we cannot assign a particular peptide to a particular protein. So they simply fall out, they go into a different database, but they show up in this analysis. Okay? So, but here we know exactly what the reason is. For the other ones, we don't. Okay? I mean, Judy, just look at here. I mean, protein ubiquitin, during ubiquitin, this is a GO category. There's a number of proteins we are not finding. Okay? So you can do the corollary analysis, low transcript level, but we detect the protein. Again, it's not everything. Again, they fall into specific classes. Okay? I don't have an explanation yet. I cannot explain to you why that is. If I could, I would probably be very excited about that. But we don't know yet. Okay? But I think the data simply point to a question here that we need to address. So how do proteins actually behave in the context of other proteins? And especially, in the cell, of course, we always think that everything is organized. Well, when we break open the cells, of course, we make a mess. Well, maybe that mess explains a lot of things, but it may also point us that in the cell, there are certain protein-protein interaction that actually affect the behavior of proteins, how then they, for example, fly in the mass spec. I don't know. I don't have a good explanation. So anyway, I'm going to stop here. This work, of course, has been driven by the dedicated people in the Genome-Investigator team. Philip Zimmermann, he's been the one who responded to the question in the lab several years ago when we were doing all this gene expression analysis. Well, can we not make an electronic northern? So Genome-Investigator, is really the result of this question. Can we make an electronic northern? And of course, the proteome analysis, the team led by Sasha Baginski, and then all of our collaborators, and many of them are actually collaborators in bioinformatics that are helping us to develop the tools that we need to analyze all the data. So with that, I thank you for listening. I'll be happy to answer questions.
Where you see different peptides coming through from the same protein in a sort of tissue-specific fashion, does that correlate at all with alternative splicing of the RNA?
Yeah, we looked into that. In one case, we could say, yeah, it might be alternative splicing, but in most of the other cases, no. It's not in regions where the protein is clearly alternatively spliced. I mean, we just have no evidence for that. So I don't know what it is. I mean, it's actually quite interesting because you see also some of that behavior in the animal work, in the animal proteomics work. I mean, we have seen similar data now. We've done a lot of proteomics not on cassava. Again, a very similar situation there if you compare, for example, cassava leaves and roots. So that is a phenomenon that has been found now in several proteome studies, and I honestly don't have a good explanation for that. Okay, so of course, I mean, the mass spectrometer is, I mean, if we are honest about it, it's still a black box. I mean, we don't know what happens when proteins or peptides fly, right? But there have been several studies have sort of addressed this question. So if you, for example, take the same set of proteins and run them again and again through the mass spec, I mean, how often actually do you see each peptide coming up from these proteins? And there's a fairly good statistical correlation what you can expect to see, but that has not. I mean, these data have not been able to address what we are seeing here. So there must be something. What I think about the protein composition. So when you extract proteins from different tissues, I mean, of course, you have a different composition of proteins. That in that composition of proteins, peptides behave differently. But what exactly it is, I mean, I can't tell you. Steve.
Willi, you said you had an enrichment of non-canonical transit peptides in the PPI2 mutant.
Well, I don't know if they're transit peptides, but these are N-termini of the proteins that don't show the typical signature of transit peptides.
The target feed would also tell you if their signal sequence is going to here, you get a small correlation.
Right. Yeah. We look for that. I mean, of course, we have any proteins that we identify that we've excluded them.
And do you see signal sequences on them, or I mean things?
In some proteins, we do. Yeah.
And the second related question, would your mass spec analysis show you glycosylated proteins? Do you see those?
We have not specifically looked for that, but I've been trying to convince people in the group to actually. I mean, Rudy Ebersol group has developed a very nice glycocapture procedure, and I said, why don't we try that out in chloroplast and see if we can capture glycosylated proteins? Because if proteins are imported via the ER pathway, maybe they're glycosylated, and it's something we need to do. We haven't done that. If you want to help us.
So if you look at your individual metabolic pathways, and you have your cases where there's a good correlation between transcript and protein, for those outliers, do you see any enrichment for microRNA binding sites or any other types of markers?
That's actually a good question. Good question. No, we have not thought about that. Yeah.
So if you look at a metabolic pathway within the plastid, what happens when you look at phosphoproteins versus all other proteins? How do your correlations change in terms of
yeah, I mean, I didn't have time to discuss that. So that's actually a very interesting question. So how do these phosphorylated proteins relate to the rest of the metabolome? What we see is that there are quite a bit of changes in the accumulation of especially when you compare end of day, end of night. Okay, so the experiments, specifically what we have done, 1 hour before the end of day, 1 hour before the end of night. Okay, and then looked at all the phosphoproteins. I was actually very shocked to see that there's very little in change in the phosphorylation of the protein. So I would have expected a massive change. It's not. But we see a number of very specific proteins. Like, for example, there's one of the subunits of the ATPase. It's phosphorylated in the dark, it's not phosphorylated in the light. It's very clear, absolutely. And then for other proteins, for many other proteins, we see quantitative differences in phosphorylation that you can also, in some cases, correlate with changes in protein accumulation. In some cases, you cannot. Okay, but for most of the proteins that we have looked at, it's not a clear, yes, you are phosphorylated or you're not phosphorylated. There seems to be, so if you have a protein that becomes phosphorylated, there might be a population of that protein phosphorylated, but then say 40% is not phosphorylated. So you see quantitative differences, but you see not yes or no. Okay, and of course one explanation could be 1 hour before before the end of night, before the end of day is too late. I mean, so we are now going 30 minutes, 15 minutes. Maybe we see more significant changes there. So we're doing these experiments right now.
If you look at the proteins which you find and compare it to those that you don't find, what is there something that comes out? So in other words, is there a population of proteins which are basically certainly at a level which cannot be tapped by this analysis? Transcription factors.
Yeah. So we looked at that, and I cannot really say that there's anything specific about these proteins that they fall into. I mean, they fall into certain gene ontology classes, but if you look at the proteins, there's nothing particular. I mean, they're not particularly structured proteins or so. I mean, there's nothing really unusual about these proteins. So that I couldn't. I mean, I would have said that if there would have or could have been an explanation.
But one takes, for example, all the transcription or the putative transcription factors and looks at them both from the point of view of expression and the point of view of protein. Do they register in the system?
Yeah, so you mean basically based on the dynamic range. I mean, if they're very low abundant, you will simply don't pick them up. Again, I mean, there is no correlation there. Okay, so I mean, I don't know what it is if you go into the very low abundant range. Okay, there are several proteins that we can identify, so we know we are sensitive enough. We can measure in the femtomole range now, right? So we are sensitive enough, but then there are in that range certain proteins that we don't pick up. I mean, we also have a problem in molecular weight range, right? I mean, we don't see a lot of proteins below 40 kilodalton. We don't see a lot of proteins above 120 kilodalton. So we're missing these classes of proteins. So we have actually, in the science paper, if you look in there, so we've actually used a specific technique, I mean, the sugar gels, to enrich proteins of low molecular weight. Okay, so I suspect actually that there's a lot of proteins out there in the 5 to 10 kilodalton range that we simply have not tapped into, and I don't know. You know, we're trying now to enrich them, but I don't know why they don't show up. You know, I mean, of course, it is again a statistical problem. I mean, if you have a protein of 60 kilodaltons, you make X number of peptides, and if you have a protein of 5 kilodalton, you get Y number of peptides, and then it becomes statistics how often you actually detect a peptide. So we're doing several experiments now to specifically go for these classes of proteins that we have not yet really identified in our analysis. So I suspect there's much more than what we have seen so far.
One more real quick question. When you take those proteins that are targeted to the chloroplast that don't have a canonical transit peptide, do they localize to the chloroplast in the mutants that you guys originally used to enrich for those proteins?
Yes. Yes, I do.
So please join me to thank Willi for such a wonderful